Cystic fibrosis is a serious hereditary recessive disease caused by various mutations in the cystic fibrosis transmembrane conductance regulator on the 7th chromosome. These mutations prevent the correct folding of a protein that serves as a chloride ion channel in epithelial cells. The misfolded proteins are never able to form the necessary passageways for chloride ions to travel across cell membranes. Improper ion flow causes the normally thin, slippery mucus that lines many bodily organs to become a thick, sticky breeding ground for infection. Scarring of the lungs is just one of the common outcomes of this disease.
An experiment was conducted to gain a better understanding of the socio-psychological implications of living with cystic fibrosis. These elements were simulated by wearing a surgical mask in multiple social settings. We hypothesized that the visual symptoms of cystic fibrosis would be perceived in a negative way by members of society because people may fear that it is contagious. We also hypothesized that someone subjected to being perceived in such a way will experience more anxiety in knowing they are being negatively judged. Tests were conducted in MSU lecture halls, an MSU cafeteria, and on a CATA bus. The data collected from each test consisted of the time it took for an unknown person to sit within a zone of inhibition predetermined as a 2-seat range to any side of the test subject from which a person can view the mask. We also quantified the increase of anxiety caused by wearing the surgical mask in a social setting using the Beck Anxiety Inventory.
For each social setting, 3 trials were conducted by each team member. The first was a control trial, which the test subject performed without a mask. The next 2 trials were experimental and conducted while wearing a mask. During the first week of the experiment, tests were carried out in Snyder-Phillips cafeteria. Meg and Jen conducted their trials at 11:20 a.m. during the lunch hour, while Ted and Ryan conducted their trials at 5:20 p.m. during dinner time. To reduce error, the test subjects sat in the same seat each trial. The classroom trials were conducted in lecture halls greater than 150 people. Classes attended by each team member included Chemistry 141, Psychology 235, Sociology 216, and Sociology 241. Error was minimized by sitting in the same seat of the classroom and keeping the sample of the MSU population constant. The bus trials were carried out on the CATA 31 bus at 10:30 a.m. Test subjects boarded the bus outside of Acres Hall and ended the trial at the exit outside of Brody neighborhood.
Tests were determined to be statistically significant when a p-value of less than 0.05 was obtained from a t-test. The increase of time for the public to enter the zone of inhibition between the control and experimental trials in the cafeteria was found to be statistically significant with the p-value less than 0.05. The anxiety scores collected from these trials were also found to be significant with a p-value less than 0.05. Statistically significant results were also achieved in the classroom trials, producing p-values less than 0.05 for zone of inhibition and p is less than 0.05 for anxiety. In the bus trials, an insignificant p-value of greater than 0.05 was obtained for the zone of inhibition test. However, anxiety scores for the bus trials were significant with a p-value less than 0.05. We predict that the p-value for the bus trials was insignificant due to the very high concentration of people in a small area. The public also knew that they will not be in close proximity to the test subject for an extended period of time. These factors may have influenced the results obtained in this section of the experiment. The results obtained from these experiments support our hypotheses. The wearing of a surgical mask in social settings increased the time it took for the public to enter the zone of inhibition of the test subject, as well as significantly raising the anxiety levels of the test subject.